I'm happy to introduce Koorosh Shahpasand, our new colleague. Koorosh joined our faculty
Recording
class
in progress.
having been recruited from the Roy Ann Institute of Stem Cell Biology and Technology in Tehran, Iran, where he was an assistant professor and the head of the Department of Brain and Cognitive Sciences. He supervised over 50 PhD and MD students, actively collaborating with numerous research groups worldwide, and has authored over 50 papers. Published papers. Koorosh received his PhD training in neuroscience from the Tokyo Metropolitan University, Japan, where he studied microtubule-associated protein tau. He then completed a postdoc research fellowship at the Beth Israel Deaconess Medical Center, Harvard Medical School, where he again conducted work focused on tau biology, culminating in a first author paper in the journal Nature in 2015. Dr. Shahpasand's current research interests focus on Alzheimer's disease and related dementias, particularly on the role of phosphorylation of tau and the conformations that this takes, and the development and treatment of tauopathies. Please join me in welcoming Koorosh for his first talk here at the University of Minnesota. So Koorosh, you can start.
Thank you. Good morning to everyone. I'm gonna talk about the central mediator of neurodegeneration, which also participates in Alzheimer's disease, a particular form of tau protein. You know, Alzheimer's disease is a multifactorial, age-dependent, and sophisticated neurodegenerative disorder with no treatment thus far. The old class medications, including like NMDA receptor antagonists and acetylcholinesterase inhibitors, are simply symptomatic and may not stop the disease development. There are two major pathological hallmarks playing part in Alzheimer's disease, including amyloid precursor protein misprocessing, resulting in beta amyloid, and senile plaques formation outside of the cells. And the second one is tau protein hyperphosphorylation and aggregation, resulting in neurofibrillary tangles formation inside the cells. Although beta amyloid are indeed neurotoxic, but it has been recently reported that tau abnormalities overlap beta with the disease development. Tau is a microtubule-associated protein and binds to microtubule structure and stabilizes the microtubules. It has like 441 amino acids, the human longest isoform, and is having like 25 phosphorylation sites. It is being moderately phosphorylated under physiological condition, but the hyperphosphorylation would result in microtubule dissociation, dimerization, aggregation, and eventually cell death. But the point is, from here to here takes quite a while to happen, sometimes over a decade. So this is of crucial importance to demonstrate which phosphorylation event would drive this pathogenicity. There are numerous therapeutic antibodies fighting with the pathological factors in the Alzheimer's disease context, including like aducanumab and lecanemab, which has been FDA approved. They are fighting with the beta amyloid, the pathogenic form. They have shown some disease-modifying effect at the very earliest stages of the disease in some proportion of the patients and have been able to slow down the disease progression. There are also some anti-tau antibodies in the clinical trial stages. Some failed, some are still ongoing. Most of them are trying to stop tau aggregation, but they have not been that promising so far. Tau is being highly expressed in the CNS, and its abnormality would result in various neurological disorders classified as tauopathies, such as like Alzheimer's, Parkinson's, macular degeneration, and etc. We have shown that the molecular mechanism of cell death is actually identical between Alzheimer's, diabetic neurodegeneration, Parkinson's disease, bipolar disorder, ischemic stroke, spinal cord injury, and etc. In most of them, tau is being the central mediator of neurodegeneration. We have previously shown that phosphorylated tau at thronin 231 could exist in various conformations. some of them are pathogenic and some are physiologic. So phosphorylation per se is nothing, but post-phosphorylation modification is of crucial importance. We have shown that actually phosphorylated tau at tronin 231 with regard to proline 232 could exist in trans conformation or even cis conformation. In the cis conformation, we have shown that the tau peptide is getting bended. That way, tau cannot bind to microtubule surface and detach from the microtubule structure and subjected for hyperphosphorylation and aggregation. We generated conformation-specific antibodies against the cis and trans p-tau and published the results actually in Nature in 2015. We initially performed the primary culture of mouse cortical neurons and put them under stress conditions. You know, neurons are very responsive to oxidative stress or nutritional starvation. So we put them under stress, and we found that stress condition would induce profound cis-p-tau accumulation inside the neurons. But trans, which is physiologic, remained unchanged during the stress condition. So it is likely possible that the factor correlates with stress at cis-p-tau, which is being accumulated under stress conditions. Furthermore, we examined the cell survival over the stress conditions. These green spots showing live cells and the red spots are dead cells. So over the time, like 24, 48, and 72 hours of stress, the cells are being degenerated like this. But importantly, if we treat the cells with anti-cis-p-tau antibody, the neurodegeneration is being suppressed because the cis-p-tau antibody could get into the cells, target neurotoxic cis-p-tau, and eliminate from the system. That way, it could stop neurodegeneration. And if we eliminate trans p-tau using respective antibody, it doesn't have any therapeutic effect in this regard, and the cells are seen being degenerated because trans p-tau is physiologic and its elimination does not have any benefit in this regard. So this is of crucial importance to detect the pathogenic tau. Tau is being served as a microtubule stabilizer factor. And as you know, neurons are having very long processes preserved by microtubule structure. So we examined the microtubule structure in cultured mouse neurons. Healthy control neurons are having very long process and actually polymerized microtubule structure. But when we put the cells under stress, such as like hypoxia, the microtubule structure is being disrupted. And because neurotoxic tau would disrupt the microtubule structure. But if we eliminate neurotoxic cis-p-tau from the system using respective antibody, the microtubule structure is being preserved just like healthy control conditions. Again, if we eliminate physiologic trans p-tau using respective antibody from the system under this stressed out cultured neurons, the microtubule are still being depolymerized and disrupted. So again, this is of crucial importance, which tau epitope to target. So what would be the physiological benefit of this? You know, microtubules are being served as mitochondrial transport tracks in the axonal cell structure. And mitochondrial transport is of crucial importance for energy supply toward the end of neuronal cell processes. So we examined the mitochondrial transport of this primary mouse culture cortical neurons, stained them with GM mitotracker, and examined with confocal over 5 minutes, time-lapse imaging. Every green spots represents single mitochondrion. As you can see over here, under healthy control condition, the mitochondrial transport is being driven very fast and efficient, something like highway actually. But if we put the cells under a stress condition, that way, the microtubule structure has been already disrupted. We found that the stress would induce profound mitochondrial transport disruption. And there is no mitochondrial transport anymore. Because the microtubule structure inside the axonal cell process has been already disrupted. So that's why the mitochondria cannot be transported. Importantly, if we eliminate neurotoxic cis-p-tau from these cultured mouse neurons, we found that the mitochondrial transport, again, is being driven very fast and efficient, just like the healthy control conditions. So the factor that playing part in mitochondrial transport disruption is likely neurotoxic cis-p-tau. And what if we eliminate trans-p-tau from this system? We found that trans-p-tau elimination would worsen the condition. The entire process collapses together, as you can see over here, because trans-p-tau is physiologic and its optional removal does not help to microtubule structure preservation. We were looking for a model to examine the antibody efficiency and actual cis-p-tau presence in the in vivo system. And we reached to this TBI mouse model, traumatic brain injury. TBI is the biggest risk factor for Alzheimer's, Parkinson's, and other neurodegenerative disorders. To induce the TBI, we employed this weight drop and weight drop onto the head of the animals. So this is the height you can see over here, and the more severe trauma would result in more profound cis-p-tau accumulation in the neurons. But there is no pathogenic cis-p-tau in the sham healthy control conditions. So cis-p-tau is likely directly correlate with the severity of trauma and stress conditions. If we claim that this cis-p-tau is the central mediator of neurodegeneration, we would have to demonstrate that it has got a prion nature and may spread everywhere, because Alzheimer's and Parkinson's, they start from one spot and it spreads everywhere. So we got to show that this cis-p-tau is also having a prion nature and may spread in the CNS as well. So we examine the TBI mouse brains at different time points after trauma. So 24 hours after trauma, we could exclusively see the cis-p-tau in the cortex, the heated brain area actually. But 6 months after that, it diffuses into deeper brain areas such as like hippocampus. So cis-p-tau is having a prion nature and may spread everywhere in the brain. Cis-p-tau not only spread in the brain area, but also it diffuses into CSF and penetrates into the blood as well. Given the fact that cis-p-tau is being appeared soon after a stress, we employed this pathogenic factor as early biomarker for neurodegeneration detection. But the concentration of cis-p-tau in the blood is actually extremely low. So we generated this early diagnosis kit. The kit is based on a golden plate, and we immobilize the antibody onto its surface. This particular structure is having an electrical resistance, and upon tau binding to the antibody, the electrical resistance of this structure would be enhanced. That way, we could detect if there is any neurotoxic tau in any samples, such as like blood, and actually how much tau is in there. Given the fact that the antibody specifically binds to pathogenic tau, it does not have any interact with blood proteins, such as like IgGs and other blood proteins. So it is very specific. Moreover, the kit is highly sensitive and can detect the very tiny amount of tau in a blood sample, almost 10 to the power of negative 14 molar. The concentration of tau in Alzheimer's disease blood sample is 10 to the power of negative 10 molar. So the kit is almost 10,000 times more sensitive than what's supposed to be. That way, we can predict if someone is going to get neurodegeneration or tauopathy years ahead of any clinical manifestation. We have published the results of this kit and are now optimizing the kit condition. This is the kit that we dropped the blood sample over here. So we are now optimizing the kit condition with respect to antibody concentration and the linker percentage. We will then examine various blood samples from Alzheimer's disease patients at different stages to quantify the key outcomes. And that way we would be able to address any blood samples to a specific Alzheimer's disease stages or even pre-Alzheimer's disease stages. The other phenomena that is being happened upon neurodegeneration is brain atrophy or brain size reduction. So we examined the brain size of these animal models, the mouse TBI models, with or without the antibody treatment. We found that while TBI induced profound brain atrophy in the animals, treatment of these animals with cis-p-tau antibody would stop this phenomenon, and there won't be any brain shrinkage, and the brain atrophy is being suppressed. So the antibody is likely having a therapeutic effect in the animal models. We examined the brain ultrastructure using electron microscopy. We actually examined the microtubule and mitochondrial structure in the axonal cell systems. You know, these yellow arrowheads showing the microtubule structure and the red one showing the mitochondria. As you can see over here, TBI would induce profound microtubule and mitochondrial disruption. But treatment of this animal with cis antibody would restore everything, and there won't be any mitochondrial or microtubule structural disruption. Furthermore, we examined the cognitive decline of these animals, of TBI mouse. To assess the cognitive function of these animals, we employed this elevated plus maze. The maze is having actually 2 open and 2 closed arms, and the entire apparatus is raised 1 meter above the floor. We are now looking from the upward, and the red lines showing the mouse movement track over 5 minutes observation. As you can see over here, the control healthy animals exclusively goes through the closed arms, and they would never go through the open arms because they are scared they may fall down or they are scared from the observer. But the TBI mouse models behave like dumb, and they show like abnormal risk-taking behavior, and they move everywhere, including like closed and open arms. But if we treat these animals with cis-p-tau antibody, again, they have an improved cognitive decline and exclusively goes through the closed arm, just like healthy control conditions. So the antibody sounds like an efficient therapeutic to fight tauopathy in the animal models. So we concluded that various stress conditions, such as like aging stress, oxidative stress, or trauma, all the same, would result in profound cis-p-tau accumulation in the neurons, and which would lead to profound neurodegeneration. But elimination of this cis-p-tau using respective antibody would stop this phenomena back to normal. However, we further examined the mouse and human tau and compared them together. And we found that there are some differences between mouse and human tau in terms of threonine 231 with respect to conformational changes. And we found that what is neurotoxic in mouse tau, the conformation I mean, does not necessarily reflect the same pathogenicity in human and vice versa. So on the other hand, we have previously demonstrated that phosphorylated tau overexpression would result in profound cell death. We overexpressed GFP tau, the wild type form, plus CDK5P25, the major kinase that play part in Alzheimer's disease, and observed with confocal microscopy. As you can see over here, GFP tau overexpression would result in profound cell death. But if we overexpress this mutant form of GFP tau, in which threonine 231 is being replaced by alanine, that way, tau protein is having 24 phosphorylation sites instead of 25. That way, we found that the same condition does not lead to neuronal cell deaths. This data is very remarkable, showing out of life 25 phosphorylation sites. Troponin 231 is of crucial importance for tau pathogenicity, likely in a particular conformation. So we perform additional set of experiments and we reach to this particular conformer, which is distinct from either cis or trans and something intermediate. We call this newly introduced conformation of tau as gauche p-tau because it's something in between cis and trans conformations. So we generated a fully human monoclonal antibody, the scFv domain, against this pathogenic human tau conformer and profiled as a US patent in USPTO. We initially performed some set of experiments. We actually outsourced this set of experiments to prevent any bias with respect to the antibody. We requested a research group in Tehran University to perform these experiments. They examined the human postmortem brain samples of healthy control and Alzheimer's disease brain extracts. And they performed a Western blotting stained with either mouse cis-p-tau monoclonal antibody and our newly developed human gauche p-tau antibody. And they found that there was no significant difference with respect to cis-p-tau amount in between control and Alzheimer's disease brain extract of human postmortem brain samples. But they found a prominent difference between Alzheimer's disease and controlled healthy conditions with respect to gauche p-tau. And our antibody could specifically bind to the 6 p-tau isoforms in the Alzheimer's disease brain extract, and it did not bind to anything on the healthy control samples. This data is very remarkable and very promising, showing that the antibody does not have any off-target in the healthy controlled brains and does not bind to physiologic tau even. Furthermore, they perform immunofluorescence staining analysis of this antibody of human postmortem brain samples of age-matched healthy controls as well as Alzheimer's disease brain samples. And they found that they stained with either cis, trans, and gauche p-tau antibodies. They found that the amount of cis and trans p-tau almost remained unchanged between healthy control and Alzheimer's disease brain samples. But the gauche p-tau is being significantly accumulated and increased upon Alzheimer's disease. That way, gauche p-tau is likely correlates with Alzheimer's disease progression. We employed this phenomena to further detect Alzheimer's disease and brain damage in the Alzheimer's disease patients. In fact, we added a histidine tag to our antibody, which is scFv domain, and radio-labeled with technetium. That way, the antibody is now a radio-labeled molecule. We initially examined the antibody penetration through blood-brain barrier, and we found that the antibody could easily pass through the blood-brain barrier in the animal models within like minutes of the injection, like 10 minutes, and accumulate in the brain of TBI mouse models. So the antibody is indeed efficient for BBB penetration. We have completed almost all the preclinical observations using the animal studies, and we are now preparing to launch a clinical trial for brain imaging of Alzheimer's disease patients in a research center in the Middle East. We will perform the imaging using CT SPECT imaging. You know, CT SPECT imaging is much more cheaper and more applicable than PET scan imaging for Alzheimer's disease diagnosis. That way, given the fact that the antibody binds to disrupted brain areas and pathogenic tau in a brain, we can detect the damaged brain area in patient brains using CT SPECT imaging. To further address if this gauche p-tau is indeed the neurotoxic factor that play part in Alzheimer's disease, and also it is different from either cis or trans, we perform additional set of experiments. We actually outsource again this set of experiments to another research center in Iran. They perform the immunodepletion process from Alzheimer's disease brain extracts and optionally removed gauche p-tau using respective antibody, using our antibody actually, and they performed a Western blot analysis. And they found that the amount of cis p-tau or trans p-tau remained unchanged upon immunodepletion of this gauche p-tau. So gauche p-tau is indeed different from either cis or trans p-tau conformer. Moreover, they performed additional set of experiments. They applied the healthy control human postmortem brain extract samples onto these cultured human neurons. These neurons has been derived from iPS cells, human neurons. So they added this healthy control human postmortem brain extract onto cultured neurons. And they found that the healthy control brain extract did not elicit any neurotoxic effect onto these cultured neurons. But importantly, Alzheimer's disease brain extract killed the neurons profoundly. So there is a factor or likely several factors in Alzheimer's disease brain extract, which is indeed neurotoxic and would kill the cells. Importantly, they applied the immunodepleted Alzheimer's disease brain extract from a gauche p-tau. They optionally removed the gauche p-tau from Alzheimer's disease brain extract and applied this immunodepleted AD brain extract onto cultured cells. And they found that this brain extract did not elicit any neurotoxicity. Again, because the neurotoxic factor which is present in Alzheimer's disease brain extract could likely be the gauche p-tau. To further address the therapeutic benefit of this antibody in the cultured neurons, we employed this iPS cell technology. We actually took fibroblasts from healthy control age-matched subject as well as Alzheimer's disease patients who are diagnosed with Alzheimer's disease and confirmed that they have like a PSEN1 mutation in their genome, in their genomes. So we generated iPS cells from their fibroblasts and generated neurons. So that way, we have real human neurons from either healthy control or Alzheimer's disease patients. So we cultured the neurons under aging stress. Normally, we are using these cultured neurons after 20 days of culture, but we culture them for additional 10 days, for 30 days. For 30 days of culturing some sort of like aging stress, we found that the healthy control cells, they remain survived. But Alzheimer's disease neurons degenerated like this. Importantly, when we treat these Alzheimer's disease cultured neurons under aging stress of 30 days culturing, but treated with our antibody, the antibody could stop neurodegeneration in these cultured neurons. And this data is very promising for future analysis, showing that the antibody could stop neurodegeneration in cultured Alzheimer's disease neurons, and likely is having a therapeutic effect in the patient brain as well. So we completed the preclinical observation of this data and gave the antibody to a clinical group to perform clinical trial phase 1. They had like an inclusion criteria and exclusion criteria. They have chosen like 12 mild to moderate stage Alzheimer's disease and perform the clinical trial. I would like to point out that the antibody half-life, according to our preclinical observation, has been 18 hours in the ex vivo conditions. So we had to repeat every 72 hours the intervention for the patients. While we were able to give them the pegylated antibody with longer half-life, They recommended to use the intact antibody with shorter half-life, but more frequent interventions. So they performed 12 sequential interventions for each patient every 3 days. So they preferred this intact antibody with shorter half-life, but they prefer this because they want to repeat the intervention for several interventions actually, for longer exposure and getting the least immunogenic response of the antibody. So they performed this clinical trial in a research center on Iran, employing 12 mild to moderate stage Alzheimer's disease. And they examined the safety analysis of this antibody in those patients. They took blood samples from the patients before the interventions, right after the last injections, and 5 months after the final injections. And they examined the safety with respect to various blood factors, such as they examined liver functions, and they found that the liver function with respect to these factors like ALT, AST, and FBS remained unchanged over the study course. Furthermore, they examined the kidney function as well, and they found that the kidney function also remained unchanged. So the antibody does not have any severe adverse or side effect on these patients. Furthermore, they examined the blood cell counts before and after the intervention, and they found that the cell blood counts remained unchanged over the course of the study. Moreover, they examined the immunogenic response employing ESR and CRP analysis of the blood samples. And they found that the antibody did not elicit any prominent or significant immunogenic response in the patients, which is promising for future analysis in the clinical trial stages. But it has to be confirmed in bigger sample size of the patients in the future clinical trial analysis. Importantly, they performed this cognitive test analysis using ICA test. This ICA test is an FDA-approved cognitive test which is repeatable and could not be affected by training. It's very friendly user, and you can choose the animate and non-animate images using an app, and it does not relate it to language or education or anything. It's very friendly user and repeatable. So they performed this cognitive test, and they found that while healthy control subject did not, with no intervention apparently, did not show any difference before and after the interventions, but the patients which received the intervention, they showed a prominent and remarkable cognitive improvement confirmed by this ICA test. Although this ICA test did not directly correlate with memory loss in the patient, but it is some sort of promising for future analysis and clinical trial studies, showing that it may have some effect in memory loss improvement as well. I would like to point out that we are now preparing to launch the phase 2A trial in a research center in the Middle East. The documents has been already submitted to the Ministry of Health, and almost they have been approved, those documents, and soon we will launch the phase 2 trial. I mean, I won't participate in that trial, but the group over there will perform and launch the phase 2 trial. And finally, I would like to thank my students who has helped me a lot in this project, my lab members and my collaborators who has helped me in this clinical trial and all the entire projects. And finally, I would like to thank you so much for your attention.
All right, thank you, Koorosh. So I think we have time for questions.
Dr.
Okay.
Bass.
Yeah, hi. I'd like to ask a question, Dr. Koorosh. Did you notice any changes in mitochondrial calcium levels after various manipulations?
Thank you for this question. Well, actually, we did not perform this analysis because this is quite new, actually, and we are at the very earliest stages of this findings. So we did not perform any analysis with respect to calcium changes in the mitochondria, but this is indeed very important, and we would need to address this.
Thank you.
Dr. Bass.
It's a great talk, Koorosh. I really enjoyed it. Couple questions. One is, with the antibody degrading so rapidly, I'm wondering if you've considered the approach of putting the antibody, the sequence of the antibody, into a DNA vector so that you're actually expressing the antibody in the patient.
Good morning, first of all. Well, thank you. Indeed, that's a very good point. But as I mentioned before, we are at the very early stages of these findings, and we are just exploring and open the window to everything. But since you asked about the gene manipulation, just let me quickly point out something. We have shown that the cis to trans conformational changes is being mediated by peptidyl prolyl isomerase 1, or PIN1. And we confirmed that the PIN1 is being downregulated in Alzheimer's disease patient brain. Because, so that way, PIN1 cannot convert cis into trans conformation. And we recently found that there are two microRNAs playing part in this disruption. Actually, there are two microRNAs that bind to promoter of PIN1 and may not allow the PIN1 gene to being expressed. So now we are thinking for some sort of like antagomers to apply onto cells and stop these microRNAs. That way we can have like more PIN1 expression and less pathogenic cis beta accumulation in the brain.
Right. I mean, that sounds really great, but I'm just putting it in your head that in terms of antibody therapy, antibodies, are proteins too. So you can get the sequence of the monoclonal antibody and you can put it in a vector and you can actually express the antibody in the patient. And that way you don't have to, give the patient the actual antibody every couple of days. That'll be, long-term treatment. So just ponder on it because it's a pretty cool kind of technology. My other question is, when you introduce the cis-P tau antibody, and it has this positive effect on the microtubules to recover the microtubules. But what is the mechanistic explanation for that? I mean, what do you think that the cis-P tau is doing to the microtubules to degrade them such that eliminating it restores the microtubules?
Well, thank you for this question. We actually thought about this because when we submitted the paper into Nature, the reviewer asked the same question. How can you explain that the intervention of this cis beta with a microtubule instability? We obviously found that pathogenic tau cannot bind to microtubule surface. And that way, the microtubule instability and dynamicity would be increased. And microtubules are very dynamic structure. And if you don't have any interrupting factor in that environment, the microtubule could be repolymerized very quickly. But we did not actually confirm this question that you asked, and we would need additional set of experiments to address this critique.
But the cis-P tau, if I understand correctly, is not binding the microtubules. right? It's not on the microtubules. So the presence of the cis-P tau, the fact that it's there, must be doing something to degrade the microtubules. And when you introduce the antibody and it binds the cis-P tau, now that negative influence is gone. So there must be a mechanistic pathway by which the presence of the cis-P tau is somehow degrading the microtubules. And, that would be very interesting to find out what that is.
Thank you. There are some hypotheses for this explanation. For example, the amount of cis-P tau in the mouse neurons are almost like 90% versus 10% of trans-P tau. And it is having like a dominating like feature. And it may convert like trans-P tau into cis-P tau. And as long as you eliminate neurotoxic cis-P tau, we perform like the seeding of this cis-P tau. And we found that when we apply the cis-P tau onto cultured neurons, the internal, the endogenous tau is also getting converted into cis-P tau. So we found that it has got like a dominant feature, and it has got like a seeding property, and may induce conversional changes from trans into cis as well. But we do not have any definite explanation for this. We have just some hypotheses, but we are exploring on that.
But you don't get that effect when you deplete tau. So it has to be a toxic effect of the aberrant tau.
Yes.
Thanks. It was really, really good. Thanks.
Thank you.
Hey, Javier, why don't you go ahead?
Thank you. Well, thank you for your talk. Great talk. I have just a quick question. What would be the relationship, or do we know already the relationship between this form of phospho-tau and the 3R, 4R forms of tau?
Well, thank you so much for this question. Well, actually, this thronin 231 or 8180 domain is actually conserved and is presented in all 6 tau isoforms, including like 3R and 4Rs. It is obvious that there are some difference between the ratio of 4R to 3R between Alzheimer's disease and healthy control subjects. Which we are also having an ongoing experiments in our laboratory at the moment. But we did not explore this one in our experimental system. But this is of crucial importance, and we would have to address this as well.
Thank you.
Sure.
Dr. Chen.
Thank you. First, this is great, fantastic talk. This is actually opens, I think, an open door for many interesting research we can do. One particular is very interesting in the part. You probably know the primary age-related tauopathy. The patient has probably just mild cognitive impairment, but invariably they all have this neurofilament tangles, tau tangles. I'm just wondering, have you ever tested the cis-P tau in part patient in the brain?
In PARP, I could
Yeah, in part, PART, the primary age-related tauopathy. Or other, you showed the TBI. Any other, could you detect the cis-P tau in other tauopathy, like a PSP, CBD, or any other tauopathy besides AD?
Well, thank you so much for this question. Yes, we performed lots of experiments and analysis on various neurological disorders. And we found that, for example, like in Parkinson's, although like the neurodegeneration start from alpha-synuclein, but it induced neurotoxic cis-P tau, and then cis-P tau spreads everywhere in the brain. And we published in neurochemistry. Moreover, we found that like glutamatergic and GABAergic neurons are being disrupted and they show like prominent cis-beta accumulation under diabetic neurodegeneration. And we published this in Journal of Neuroscience as well. So we have, importantly, we found that cis-beta is also being accumulated and appeared in the spinal cord injury even. And the spinal cord injury, which did show like a prominent amount of cis-beta accumulation. So we examined several neurological disorders, including like ischemic stroke, as long as I remember, bipolar disorder, macular degeneration, and etc. Yes, we confirmed that.
So in other words, in those disease or lesions, you may not see tau tangles, but you can detect cis-p-tau if my expression is correct. Because in those lesions, we may not see like mature neurofibrillary tangles.
Well, you're right. This cis-p-tau is a monomeric factor, and at the very early stages. And I'm not sure how could it be converted and accumulated into neurofibrillary tangles or not, because we did not perform this analysis. But we found that apparently in Alzheimer's disease, neurofibrillary tangles are being formed, and this pathogenic tau is co-localized with neurofibrillary tangles. So in Alzheimer's disease, we are pretty sure that it accumulates, but we did not examine like the tangle formation in other parts.
Interesting. Thank you. It's great.
Sure.
Great talk.
Thank you.
All right, any further questions? If not, we'll close for the day. We do have APCP clinical faculty meeting at 9 o'clock, and we'll start right on the button. Thank you, Dr. Shahpasand. That was wonderful. Have a good day, everybody.
Thank you. Thank you.